So we will move next to the main event of today's. So I will pass this time to our moderator, Dr. Wawaimuli Arozal, M.Biomed, Ph.D. So please, Dr. Wawaimuli.
Okay, thank you very much, Dr. Retno. So good morning to everyone. Good morning to every participant and also our honorable speakers. Welcome to our webinar session entitled Recent Breakthrough in Neuroscience. So I am Dr. Wawaimuli. I will be facilitate this session. So today we have 3 speakers. So the first one will be Professor Teruna, and then Professor Hee Jae, and the last one is Dr. Satriotomo. So I will inform the regulation of this session. So after complete the speaker one, the first speaker complete presentation. My voice is clear right now?
Yeah.
Yeah.
Okay.
Yes.
Yeah, Doctor.
Okay, so I will inform the regulation of this session. So after finish the first speaker, so all participants can directly ask the question to the speaker or by chatting. So you can show the question either by English or bahasa. So maybe with I will start this session with I invite the first speaker, Professor Teruna Siahaan. So before his presentation, I would like to share his CV. Okay, everyone can see?
Yes.
So Professor Teruna Siahaan, Professor Ayat Takeru Higuchi Distinguished Professor. He was born in Medan, and then now stay in Kansas and work in University of Kansas in the United States of America. So the education background of Professor, he was graduate from chemistry in University of Indonesia, and then complete the master degree in organic chemistry also from University of Indonesia, and then complete Ph.D. degree from University of Arizona, and then continue for a postdoctoral program in University of California. So he has a publication. I noticed 200 publications, international publication. Now he's director and global health and research school of pharmacy in the University of Kansas. So the research interest of Professor Siahaan is only utilization of cell adhesion molecule on the cell surface of enhancing drug permeation through the intestinal mucosa and blood-brain barrier. So I think this is all the briefly curriculum vitae for Professor Teruna. So without further ado, I would like to invite our honorable speaker, Professor Teruna, to start this lecture. Please.
Okay. Okay, thank you. Thank you very much. You can hear me, right? Okay.
Yes.
Okay.
Yes.
All right. So I'm going to share screen. Okay. Hopefully you can see my screen, right? Okay. Thank you for the nice introduction. Good morning, everyone. So my topic today is I'm going to talk about peptides and proteins, potential treatment for brain diseases. So as you know, there is so many problems in treating brain diseases such Alzheimer's, multiple sclerosis, and brain tumor, including Parkinson's diseases. So part of the reason is that it's very difficult to deliver molecules to the brain. And until now, there is no cure for MS or Alzheimer or Parkinson's diseases. So most of the drug that you have is actually only suppress the symptoms, so you cannot reverse them. So nowadays in US, many efforts been done to treat brain diseases using neurotropic factors and monoclonal antibody. For example, in Alzheimer's and multiple sclerosis, people try to deliver antibodies to the brain to treat those two diseases. So as I mentioned before, one of the problem is in the difficulty to deliver this molecule to the brain. So if you think about antibody or neurotrophic factors, especially protein, it's very difficult to deliver them to the brain. Even small molecules cannot get into the brain very easily. So this is part of the problem in treating brain diseases. The reason is like you have, you have this what we call blood-brain barrier. So you can deliver the molecule to the bloodstream or to the systemic circulation, but they cannot pass the blood-brain barrier because of the protective nature of the blood-brain barrier. So one of the things that I'm going to talk about that we did in our lab is we are looking into multiple sclerosis. Multiple sclerosis is actually it's autoimmune disease that in which immune cell infiltrate the brain and start attacking the axon, and then damage these myelin sheaths. Therefore, signal cannot go across the neurons. So as the effect of this, you can have the patient becomes paralyzed, so becomes disabled. So worldwide, there are about 2.3 million people suffer from this, and we don't have cure. Most of the drug that we have, they mostly halt the disease progression. They cannot reverse them, and usually they're difficult to diagnose. Now, cost per patient is quite expensive. It's about $60,000 per patient. So we have a lot of drug for MS, but most of this drug only suppress the symptom. They cannot reverse. For example, you cannot really remyelinate the brain, so only neurotrophic factor probably can do that now. So people cannot reverse the disease. Maybe you can halt them. There's drug called Cybri. Cybri is an antibody. It's antibody that inhibit cell adhesion. So when immune cell try to infiltrate the blood-brain barrier, they adhere to cell adhesion molecule called VCAM. So the Cybri, what it does is bind to one of these proteins. So they inhibit infiltration of immune cell into the brain. So Cybri works actually in the bloodstream. So one way to reverse this is by regeneration or remyelination. So therefore, people are interested in delivering molecules that can do that. So companies in US develop many antibody. This antibody actually can remyelinate the axon, but during the clinical trial, most of these antibody not successful to pass the clinical trial. So they fail in the clinic. Then the alternatives, people thinking about neurotrophic factors such as BDNF, NGF, and IGF-1. So in our study, we are thinking about BDNF because it's well studied and people have been working on this for a long time. So that's the topic that I'm going to talk about, how we deliver BDNF to the brain. So the other disease that we are trying to do, and we is looking into Alzheimer's. So again, this is A lot of people suffer from this, and majority of them 65 or older, especially in US. The lifespan is getting longer and longer, so many people get diagnosed with um, Alzheimer's. So many efforts in US to try to treat this disease. So basically, what happens is you can see the brain kind of like deterioration of the brain and Alzheimer's. One of the hypotheses is that the Alzheimer's brain, there are two hypotheses. One is the uh, tau tangles and the amyloid plaques hypothesis. So as you can see, people with Alzheimer's, you can see the plaque and the tangles. So there are efforts in this area. People deliver antibody to beta amyloid. And as you can see the next, there are many efforts that fail. Current treatments for this basically managing the symptoms using acetylcholinesterase inhibitor and also NMDA receptor regulators. So many companies try to look at ways to reverse the disease, and they develop anti-beta amyloid, but this antibodies, there are 7 of them fail in the clinic. Recently, a company has claimed that adenosumab has some efficacy in Alzheimer's patient, although the approval is still pending, so the jury is still out on this. So from our perspective, is one of the problem here is the difficulty to deliver this huge molecule. Antibody is 150 kD. Even small molecule cannot go past the blood-brain barrier. So if you think about large molecule like this, it's not as easy to get into the brain. Although there is hypothesis that antibody get into the brain using the Fc receptor. So one of the thing with antibody is that they are quite stable in the blood. The reason is they bind to Fc receptor, they get protected and get into the cell, get protected from a lot of enzyme in the blood. So that's one of the reason a lot of company working with antibodies. So the other alternative, as I said, same with MS, the possibility of delivering BDNF to the brain is one of the things that we can do to solve the problem. So BDNF is actually a dimeric, and it's about 13 kD as a monomer. They work as a dimer, and they activate CREB signaling. And then one of the hallmark of this, when you deliver BDNF in the brain, and then you can see for correlation of TRKB. So the other thing that you can check if you are successful in delivering BDNF, there are other marker like ERK1 and ERK synthesis, so you can detect this. So in our work in the future, as you can see, we can look at this marker to see if we deliver BDNF. So people have treated actually MS by delivering BDNF via ICV. Now, obviously, this is pretty invasive, so it's not convenient. Obviously, patient won't like to go through this process because you have to drill the skull and then deliver them into the brain. It's kind of dangerous. So if you can find a way, a more comfortable way to deliver the molecule by delivering them via intravenous injection, then probably it will be helpful to the patient to do this. So our efforts in my lab is try to deliver proteins, including BDNF antibody and other proteins, via IV or via carotid artery here, and to improve the delivery of this protein into the brain. And we use animal model for MS and Alzheimer's, also brain tumor, to solve that problem. I don't have time probably to talk about the brain tumor, but I'm going to focus on the BDNF on animal model for MS and Alzheimer's. Okay. So for those of you familiar with this, how do you overcome the blood-brain barrier? So basically the blood-brain barrier is the microvessel that goes through your brain. And if you can see this, there's a endothelial cell forming this microvessel, and then you can see the junction. So our focus, our work is focused on this junction, basically. How can we let molecules percolate through this junction, penetrate through the junction without opening the junction in a big way? So if you look at the cross section, most molecules can pass through the junction through transcellular pathway by partition through the membrane. But big molecules like protein or peptides even cannot go through transcellular very easily. Now, we can go through this paracellular through the crack here, but even then it's very tight. So for that, if you ever heard about Lipinski rule, if for molecules to pass through the transcellular, they have to have certain physical chemical properties, usually molecular weight less than 500 Dalton. So if you can imagine, antibody is like 150 kilodalton and has to have a log P less than 5, and the hydrogen bonding donors less than 5 and 10 for acceptor. Now, molecules, like I said, antibody has been proposed to go through the transcellular by receptor mediated by Fc receptor. So that's one of the proposal. The other thing is that the blood-brain barrier has this efflux pump, so it can pump the drug that tried to penetrate the membrane back to the blood. So that's the reason a lot of anti-cancer drug, a small molecule, cannot get into the brain because it is a substrate for the efflux pump. So it's quite difficult to deliver this molecule. So how do we increase the porosity of the paracellular pathway, which is the junction between endothelial cell? So that's the idea. So normally molecules that bigger than 11 angstrom cannot pass through this junction because there's this what we call a tight junction. So what we are trying to do is how can we open this junction selectively or reversibly so we can let molecule pass through this junction. So in this case, they cannot pass through the junction. One way to do this is we are developing a molecule that can inhibit this zipper. We call it zipper. This is a zipper or Velcro. So we develop peptide that can bind to this molecule so they can open the junction temporarily so the molecule can penetrate through the junction and get it into the brain. So that's our idea. So to make a long story short, we found two peptides that we do this job. We call it HAV6 and ADTC5. This is a linear and cyclic peptide. It comes from this protein, the junction protein that you see that I show you as a zipper molecules. So there's two peptides that we can that we discovered to do this. So this is an in vitro assay that we use. We make a model of a monolayer of a barrier. In this case, we have MDCK. When we add the peptide and the electrical resistance drop, and then when we wash and replace the media, they can go back up. And then as a positive control, we use EDTA. EDTA is a calcium chelator because the junction protein is calcium binding protein. And then they drop the resistance because ion can pass through the junction because it's open the junction. And then when we wash again and replace the medium, they reseal. So that's a reversible process. So this is what we hope because we don't want to open the junction too long. It has to be reversible. So to test the idea, we also tried to deliver mannitol. Mannitol is a paracellular marker. They pass through the junction. So this is a control mannitol alone. But if you put our peptide here, you can see 8 times enhancement. If you modify the peptide, just one residue, you can increase the activity of the peptide to modulate the junction about 17 times. So how about in vivo? Can you prove this in vivo? So we've done this many times. We use MRI. So we use MRI enhancing agent, GDTPA. If we incubate, we inject the animals with a mixture compared to just GDTPA alone. We can see the brain scan. So here, if you can see at time 0, this is a control, and then this is with the peptide. Then after 9 minutes, you can see in the mixture between the GDTPA and HAV peptides, a lot of them in the brain here. So we can quantitate this, and then we can see here the quantitation. After 3 minutes, you can see GDTPA a lot in the brain, and then it will be cleared. And then if we inject GDTPA again, and it will go up. So the opening up to about 45 minutes. Now, we know this because what happens is we did the experiment, we inject the peptide, we open the blood-brain barrier, and then we wait 1 hour and we inject GDTPA. No GDTPA can get in. It means the process is reversible. About 45 minutes, they open the blood-brain barrier. So, so here we said, okay, we can deliver MRI enhancing agent. How about protein? So in this case, we are interested in delivering BDNF. For proteins, actually, we use ADTC5 peptide, another peptide. Remember, I told you we have 2 peptides. So, so first we deliver, we have a marker BDNF. This is just BDNF, and then we deliver BDNF alone to the via IV. As you can see very much BDNF in the brain. This is using Western blot. Okay, so and then if we increase the dose and then deliver them together with ADTC5, we can see BDNF that we deliver in the brain. Now, if we increase the dose of BDNF to be higher, then we can see quite a bit of BDNF in the brain. So we got quite happy about this. And actually, with other proteins, we've been able to label the protein with NIR dye, I'm sorry, and then we deliver them with and without BDNF, and I'm sorry, without ADTC5. So with ADTC5, we can see decoration or this deposition of the protein in the brain. And without ADTC5, we see very little in the brain. So the other marker for function of BDNF when we deliver them in the brain is phosphorylation of TRKB. So as you can see here, the first 3 we deliver BDNF alone. We don't see any TRKB in the brain, phosphorylation of TRKB in the brain. And then we deliver a low dose and we can see some TRKB. When we increase the dose, we can see a really pronounced TRKB phosphorylation in the brain. So we are quite happy about this. Then we say, well, what would happen if we deliver BDNF alone and then with ADTC5? Can we treat the disease? So our model now is multiple sclerosis model. We call it EAE. This is an accepted model for MS, try to evaluate drugs. So what usually we stimulate the disease using what we call it PLP. This is myelin sheath. And usually when we stimulate with PLP, you get this relapse and remission model. It means the animal will exacerbate and then it will remit and then relapse. So when you stimulate the disease, what happens is the tail becomes weaker, and then as the disease progresses, you can see the hind limbs become even weaker, And then finally, when you have score of 5, all 4 limbs paralyzed and the tail is also weak. So what we say, okay, what would happen if we stimulate the disease at day 0, and then at day 9 they goes up, and at day 13 they are high, the peak of the disease. Now usually you got remission at day 21 to day about 40 or 45, and then a relapse again. So our goal is when they remit, we treat them with BDNF alone and BDNF with our blood-brain barrier modulator. So we treat them every 4 days, a total of 8 times, okay, to see the effect of our modulator to deliver the BDNF. So we have 3 arms. One is actually 4 arms. One is PBS alone, which is a control, and then we deliver our modulator alone, and then we deliver BDNF alone, and then we deliver BDNF with our modulator. So as you can see, we can look at the score. You can see here the exacerbate and then remit. And the first one is the vehicle. You can see they relapse back, and then this is we integrate Irawan under the curves just so we can quantitate the disease score. And then we, when we deliver our modulator alone, and the disease is still the same, it relapse. And then when we deliver BDNF alone, it still relapse. The reason BDNF alone, as you can see before, even though you inject them IV, they cannot get into the brain. But when we delivered BDNF with ADTC5, you can see they don't relapse. You know, after remission, they keep going down, the score keep going down. So this is quite um, quite good, and we're quite happy about this to see the effect of delivering BDNF via IV. So we have a way of doing a lot gentler way to deliver a drug to the brain. So then the question is, does at the end of the day, the BDNF induce remyelination in the axon? So here, the first part here is a vehicle. So what we are seeing here is we are quantitating the damage, which is the white spot here. So the damage is the demyelination quite high. And then if we deliver BDNF alone, it's still quite high. But the animals that treated with BDNF and ADTC5, they quite a good remyelination in the brain. So then the question is, normally remyelination is generated by oligodendrocyte precursor cells. So normally people look at the NG2 receptor. So the cell has NG2 receptor, so we can do antibody binding to that cell, to the NG2 receptor. So this is a negative control. And then when this is the brain of animal, in the corpus callosum, we look at that get treated with vehicle, not much of NG2 there. And then the one that got treated BNF alone is pretty low. We were surprised that to see that the one that treated with BDNF and ADTC5, we can see lots of brown spots of NG2. So we quantitate the pixel, quite significant increase in the NG2. So the next we try to do in situ hybridization to look at the AGR1 ARC and NO synthase, because this is a marker for neurodegeneration. So this is kind of interesting. There's a new probe that people use. They have this ZZ probe, so they have less, they're more sensitive and less false positive. So when we look at the 3 different arm vehicle, BDNF alone and ADTC5, we look at the mRNA of EGR1, ARC, and including NOS. So when we treat the just the vehicle treatment, we see a low, like a standard AGR1, and then the BDNF alone is almost the same. But the brain that got treated with BDNF and ADTC5, we can see a high amount of AGR1. The same thing with ARC, the control and then BDNF alone about the same, and then quite significant increase in the ARC1. The other thing we try to look at the NOS, and then there's no difference in that. So with this, we're quite satisfied with the data that we have to see that that BDNF can be delivered, and then they have function in the animal model. Next, we said, well, we want to do the same thing, but we gonna use APPPS1 Alzheimer's disease model. So this is we let the animal becomes to get Alzheimer's after 6 months, and then at the end we treat them with BDNF. So 6 month age of APPPS1 animal, and then we have 3 arms, and then we inject them every 4 days, 8 injections. Similar treatment as the MS, BDNF with ADT and BDNF alone and vehicle. So then what we did is we are looking at the cognitive improvement of the animal using Y-maze and the novel object recognition. At the same time, we look at the NG2 cell in the brain, and then again, the neuroregeneration marker. So for those of you who's not familiar with the Y-maze test, so there's a Y-maze like this, like a Y. So what you do is you close the first arm, you get the animal to get familiarize themselves with the 2 arm. And then we open the arm, and then we monitor using video and to see if they explore the other arm. So after we got the baseline, we look at the treatment. So looking at this, we compare 3 arms. One is the animal that is treated with vehicle alone, and then BDNF alone, and then BDNF with ADTC5. So here we look at the percent time spent in the 3rd arm when we open them, and when we have the vehicle compared to BDNF alone, they are almost comparable. But when we have we open the 3rd arm, when we look at the animals that treated with BDNF and ADTC5, we can see the increased time spent in the 3rd arm. So indicating that this animal has better cognitive function than the other 2 groups. And we also we look at the number of entry, almost similar to the percent time spent. The BDNF alone and vehicle, they are almost the same. And then the BDNF with ADTC5, they are a lot more number of entry in the open arm, the new arm. Next, we look at the novel object recognition test. So in this case, we are familiar with the mice with the 2 same object, and then after a while, we change one of the object. So if the animal presumably has a better cognitive function, they will explore the new object. So the more they explore the new object, they have a better cognitive function. So here is the data. The vehicle, we have low curiosity with the new object, and then for the BDNF, slightly better. But when we have ADT C5 deliver the BDNF, we got quite significant time interacting with the novel object. So we look at the total time interacting with both object is almost the same, except the animal that treated with BDNF and ADTC5, they are more curious and they look at the novel object more than the other 2 groups. So, and then we try to look at, does BDNF change the amount of plaques in the hippocampus? We know in the literature there is no effect on this usually, and then we confirm that we don't see the change in the plaques in the
Kalau yang
hippocampus.
saya ingat ya dok ya, dia itu ini kemarin tuh apa sih, makan bakso gitu loh sama kakaknya.
Can somebody mute that?
Jajan bakso gitu sama kakaknya gitu. Sebelumnya Mbak,
Can somebody mute?
belum sih dok ya, baru kesini aja, baru ke dokter ini.
Can somebody mute the?
Yes, just a moment.
Okay, thank you. It's kind of distracting,
Okay.
so we notice, and then there's no effect in the on the plaques. So how about the NG2 cells? So we look at just like in the MS model, we look at the upregulation of NG2 cell, and the vehicle group and the BDNF alone, we didn't see any enhancement in the NG2. But when we look at the BDNF and ADTC5, you can see the increase in NG2 cells in the brain. Similarly, we are looking at EGR1, ARC, and then MAPK. So as in the MS, we can see that vehicle and BDNF alone is similar, but when we treated them with BDNF and ADTC5, we can see an increase in the EGR1 and ARC, but we don't see any difference in the MAPK. So after this, we are interested in looking at antibody because we are working with a company that asking question, can you really improve the antibody? For example, beta amyloid antibody is kind of fail in the clinic. Maybe we can improve the delivery of antibody. So we've been working on antibody, and I don't have time to show you a lot of data here, but antibody, as you see, is 150 KD, which is pretty huge, bigger than BDNF. So here we have 4 animals injected with the antibody alone. So basically, the antibody is conjugated to near IR dye. So there are 4 animals here. You can see very faint amount of antibody in the brain. But when we deliver them with ADTC5, this is, you can see the enhancement of antibody in the brain. The movement there is actually about 8 level of scan of the brain using near IR fluorescent scanner. So it's about the increase is about 4 times. So the experiment is actually we inject the antibody alone or the antibody with our modulator, and then after 15 minutes we sacrifice the animal and then scan the brain. So it's pretty short. In a short period of time, you can see the increase of the antibody in the brain. So the last part, maybe about 5 minutes, looking at question, how do this peptide works? How do they modulate the interaction? So we would like to know the mechanisms. So as you know, we can study the binding site of the peptide to the protein using NMR. So we use this Hsqc Nmr. So what you're seeing here is the connection between the N and the nitrogen and hydrogens of the protein. So we express the protein with N15 label. When we titrate the protein with the peptide, then if the peptide bind to one side, therefore the NH chemical shift will shift. Like for example here, the serine 78, you can see it was here before. As you increase the concentration of peptide, because it bind to serine 78, they move because the peptide sealed the nuclei from magnetic field, so they shift to the right here. So that way we can combine the SIFT and molecular modeling, so we can determine where the peptide bind. So we, as I mentioned before, we have two peptides. One is HAV6 and ADTC5. We have done this study that we know the binding site is quite different. So then we know that the binding site of ADT peptide here and the binding site of HAV is here. So we look at the model of cadherin. So this is X-ray structure. So actually it's interesting. Cadherin bind, this is the membrane 1, this is membrane 2, has 5 domain. So there's a cis binding between two cadherins, so using EC1 and EC2. Now the trans domain bind to EC1 and EC1. And the EC1 and EC1 has what they call it swapping domain. So they latch to each other using tryptophan residue here. So looking at that and looking at our peptide binding sites, is actually our HAV peptide bind to this region, so dissociate the cis interaction. So as a result, our zipper molecule looks like this. From here becomes like this. That's the reason probably it's opened the junction, not completely, but increased the porosity. Now, the interesting thing is the ADTC5 bind to this domain swapping. So there's no domain swapping. So what happens is it break this bond here to form, to open the junction. So that's the reason probably ADT is a lot stronger in opening the junction than the HAV. So actually this process is reversible. And for small molecule, basically, like I told you before, for HAV peptide, it can open the junction for small molecule, but after 45 minutes, they close. Now, for ADTC5, it will open about 2 hours, but close again. Now, if you increase the size of the molecule, the opening is for that large pore is shorter. So we have a hypothesis that our modulator is open large pores, and then they soon collapse to a medium-sized pore, and then the medium-sized pore collapse to a smaller pore. That's the reason for smaller molecule, they can penetrate longer into the brain than the larger molecule, which is kind of good because we don't want large molecule get into the brain easily. So as you can see, we've shown that we can deliver BDNF. Actually, we deliver other things also, but for the sake of this talk, we have shown you we can treat, reverse at least the disease symptoms or remyelination of the brain in MS model, and we can look at neurodegeneration in Alzheimer's model. So we actually have data to show that we can deliver small to a large protein up to antibody, but we cannot deliver molecules larger than 250 kD. So there's a limit size there. So we've shown that ADT is better than HAV peptides. And then currently we are optimizing this so we can efficiently deliver antibody to the brain. So I have a lot of students working on this. We're supported by NIH, and I'll be happy to answer any questions. Thank you.
Okay, thank you very much, Professor Teruna, for the nice presentation. So we are very happy. I mean, very interesting presentation related to attach the BDNF to the ADTC5 to improve the pass the kinetic of BDNF to the barrier. So there are a lot of questions from the audience. So the first one, maybe I will select a couple of questions. The first one coming from Nul from University of Brawijaya. So he asked, based on your experience, which one has more potential to develop? Some active compound with some target, or one molecule for certain target? As we know, for degenerative disease is multifactor causes.
Okay. Yeah. So I guess I like to deliver molecules that I know the mechanism, and so I don't have any problem if people deliver mixture or different molecules or 2 or 3 different molecules at a time. But the question is, do you know the mechanism? Each one of them probably is important also, because as you know, drugs usually has, even though they have benefit, but sometimes they have toxicity. So we have to kind of balance the two, right? So it's important to know how the molecule works first. When we are in industry, now for cancer, usually we do screening, but at the end of the day, sometimes we like to know the mechanism so we can handle it just if we see some toxicity, and then we can modify them maybe to avoid them. So I have no objection in having mixture, but it's better if you know the function.
Yeah, one molecule is better, yeah, Professor Teruna.
Yeah.
Yeah. Okay. The next question comes also from Norma. So he asks about the herbal medicine that we call jamu. So you know that in Indonesia we develop many kind of jamu. So jamu maybe has a potential as a cure for the neuroscience field, especially in dementia. What your opinion about that?
Yeah. So people are studying curcumin, for example, right? Curcumin is from kunyit. Is that kunyit, right?
Yes.
Yeah. So one of the problem with actually there's a clinical trial people done in Italy with curcumin, but for Alzheimer's, but the clinical result is negative. But it doesn't mean it doesn't work. It could be. I know if you know curcumin is a pretty hydrophobic molecules, right? So it could have a solubility problem.
Yes.
All right. So you have to kind of like deal with the solubility.
Yes.
And then the other thing is we don't know, and people show that, I think before that clinical trial, people show in the animal model, they pass the blood-brain barrier. Now the question is, what dose do you need to pass through the blood-brain barrier to have an effective drug, right? Efficacy, I guess. So one of the problems, as far as I know, curcumin has a problem with solubility.
Solubility.
So people need to kind of look into way to do that. Now people try to formulate them using nanoparticles, using liposome. Now, if you imagine nanoparticles, is very hard for them to pass the blood-brain barrier. Even small molecules, difficult. But if you find a way to do it, and now people use microbubble using ultrasound. So they use particles like almost like my cell, but they improve the delivery using ultrasound. Now they show in the animal model they have efficacy in neurodegeneration in the brain. Now, what is the effect of those ultrasound, repeated use of ultrasound in your brain? I'm not sure. So.
Okay, thank you very much, Prof. Teruna. So another question from Dr. Martina. So she asks about the herbal also that claim have BDNF, a lot of BDNF. I don't know what kind of herbal. So it is possible that herbal can buy orally, then effect for the neuroplasticity of the brain.
Yeah. So if you it's hard for me to believe that even though the herbal has BDNF, in our study, BDNF alone is not going to work. You cannot pass through the blood-brain barrier. So when I start my work here at KU in '91, which is 29 years ago, a lot of funding by NIH to support delivering BDNF into the brain, and all fail. So without success. So NIH after 5 years stopped those funding. There is no funding because nobody able to do that. So that's the reason we are using BDNF as our marker, because people are interested in doing that. Now, it could be that the herbal molecule in the herbal maybe a small molecule that stimulate generation of BDNF in the brain. That could happen. But if it has BDNF alone, I cannot believe that. But it could have a small molecule that can penetrate the blood-brain barrier and stimulate the production of BDNF, because we produce BDNF in our brain, right? So in fact, people have studied that for Alzheimer's patient, if they exercise, so when we exercise, we generate lots of BDNF in the brain, right? It's been studied here that Alzheimer's patient that exercise, their cognitive function is better than the one that's not exercise. And they've done in animal study, they see the increase in BDNF when you exercise. Now, it could possibly that the herbal has a small molecule that can trigger the production of BDNF, but I cannot believe it has BDNF that can pass through the blood-brain barrier. It's hard to believe.
Yeah. Thank you very much, Professor Teruna. So we understand the point. So I mean, the other question from also from the speaker, Prof. Lee. So happy birthday to you. This is your
tomorrow in the US is my birthday.
Okay.
Well, here is still November 30th. So tomorrow is December 1st. Yeah.
Okay. Happy always.
Thank you. Thank you.
So Professor Lee asked the question about the role of a blood-brain barrier in preventing the pathogen in the brain. So the question is, after you treated with ADTC5 modulator, did you see any activation of microglia? And also, how about the other inflammation factors?
Yeah. Very, very good question, Dr. Lee. Yes, we've done this before. So it's always happened if you modulate the blood-brain barrier. It could happen that you have inflammation. We did animal study and we didn't see any astrogliosis and inflammation in the brain. Now, I know if you're familiar with Dr. Ed Newalt. Ed Newalt is the person who deliver anti-cancer drug to treat brain tumor patient using hypertonic mannitol, which is they call it osmotic delivery. So what he does is he injected anti-tumor drug through the carotid artery here using 30% mannitol. So hypertonic solution will shrink your cell, right? So it shrink the blood-brain barrier, and it's actually open the blood-brain barrier non-selectively. So let molecule pass through. And in his study, usually has a side effect. The side effect is inflammation and astrogliosis. So, but in our case, we don't see that. We look at animal model. We didn't see any of them because our method is modulating only one fraction of the Velcro. So if you look at the structure of intercellular junction of the blood-brain barrier, there is occludin, claudin at the top. We got nectin and cadherin, and we've got desmosome at the bottom. What we're modulating is only a fraction of the adherent junction at the bottom. So although they are connected to the top through Z01, catenin, it's open, but it doesn't open like when you shrink the cell using mannitol. So the short answer is we didn't see those two, inflammation, astrogliosis. Yeah. Very good question.
Okay, thank you. So if we have any time, we still have time. So
I think so.
Okay.
Maybe. Yeah.
So next questions coming from Suharti. So she asks about the mechanism of ADT5 unique BBB permeable, and it is only permeable for specific to BDNF or what else?
Okay. Yeah. So the use of the ADTC5 or HAB peptide, we use it as if you want to call it like adjuvant, almost like adjuvant. So if you have any drug, any molecules, you just mix them together. We don't have to do chemistry. We just like mix them together, because the function of the peptide, the ADTC5, is just to open the junction. So if you have high concentration of drug that you want to deliver in the blood, they will percolate, penetrate to the junction. Now, because of this binding, it's just protein interaction, so it's like an equilibrium. Now, sooner or later, your peptide will be cleared. So when it's cleared, it will reseal the junction. So that's the reason we see only 1 hour for ADT for small molecule, and then 2 hours opening for ADT. For HAV is 1 hour, less than 1 hour. For ADT is 2 hours, because they sooner or later they got degraded. We don't want them to open forever, because it's kind of dangerous. Yeah, just like Dr. Leah question, you can generate inflammation in the brain because other protein might get into the brain. So we don't want that. We want just so that's how the criticism, part of the criticism in our method is because we are playing with the blood-brain barrier. It's kind of dangerous. But as you know, anything that you do, you have to kind of like know exactly how to do it, right? So you fine tune it.
So the ADTC5 can be purchased commercially.
You can make it yourself.
Oh, we can make by ourselves. I mean, manual.
We synthesize it ourselves.
Oh, okay.
Yeah, people who
So-
know how to synthesize peptide, they can make it.
Oh, I'm not, I don't know about that. Okay, so next question from Gallery. So he asks about, is there any latent waste time after 60 minutes of ADT5 administration on animal model? Or what is the recommended interval in first and second administration in the latent phase?
Okay.
And then,
Yeah. So like I said before, so let's so it's different molecule, different size of molecules. The opening is different, the length of the opening. So when we, for example, let's look at ADTC5. So if you deliver small molecule, right? ADTC5 can still penetrate the brain after 2 hours incubation. Okay, but for albumin, for example, which is 65 kD, after 20 minutes is close. For antibody, if you wait 10 minutes, it's already close. So depending on the size of molecule that you deliver. So for antibody, if you deliver the ADT and then wait 10 minutes, no antibody get into the brain because it's close already. The size of the pore is already close. Now for small molecules still penetrating, that's why the last mechanism that I show you, that they are kinetics of collapsing of the pore. So that's the one that we are studying right now. How long, how big? Yeah, we have to fine tune that.
okay, sorry. Next question from the same participant. So he asks about the effect of on the tau protein, Professor.
Okay, so right now, I don't think that we don't know if have any effect on the tau protein, but right now, I mean, most drug that's designed for Alzheimer's, I think the one that's very advanced is the antibody for beta amyloid. Now the tau protein, I think inside the cell, so it's a lot more difficult to deal with protein right now. You have to have other ways to do it. So people try to deliver enzyme inhibitor for secretase, try to prevent degradation of beta amyloid, APP to beta amyloid. The most advanced one right now is just antibody to beta amyloid. Antibody to, yeah. So we are working on that right now with a company, try to see if we can improve the delivery. So, to
So
kind
now,
of like
clinical trial setting, Professor? the
No,
antibody?
no, no. The antibody, yes. The antibody, there are 7 antibody already fail.
Oh, okay.
There is one that said after looking at the data, they see some good efficacy, and but FDA is still kind of like pending approval because they didn't see too much of efficacy.
Oh, I
Yeah,
see.
so
It's promising, yeah.
still promising, yeah, but remember it's very expensive treatment.
Okay.
Yeah.
Yeah, the problem. Okay. Next question from I will select only the different question. So from Amalina, he asks about the aside from microtubules that you mentioned earlier, what about the exosomes, peptide masking or modification, the size molecules such as nanoparticle? Is it possible to deliver the drugs to the BBB?
Yeah. So like I said before, people look at exosome. There's a person in Australia, I think, focus on exosome. So exosome is probably promising because they might get into this endothelial cell and then exit on the other side into the brain. But there are methods that people use, for example, transferrin receptor antibody. So people look at transferrin and bind to the receptor, and because transferrin receptor is high expression in the blood-brain barrier, but what they see is not much molecule. The antibody can't go into the brain side. It can go to the brain side, but the antibody never release from the receptor, so they cannot really effectively diffuse to the other part of the brain. So that method is not that efficient right now. So going into the cell using nanoparticles might be trapped in the lysosomes. The same thing with the antibody that bind to transferrin receptor. They got degraded in the lysosomes. They never passed through the cell into the other side. They did pass, but after pass is not very efficient, and then they're going to get released and then diffuse to the site of action. So it's a difficult problem. I mean, this is the area of research that you guys should look at. it's a flush with questions to do.
Yes. Okay, thank you. Maybe this is the final question, Professor. Yeah. So someone asked about the ideal time to check the Y maze for the rat. Maybe we can also perform in neonatal rat. Is it possible or not?
I'm not too familiar with the neonatal. To be honest, we collaborate with experts of Alzheimer's disease to do this. So we use just our animal APP model and then triple transgenic model that's already like 6 months old. So I cannot answer that question. But the Y maze is quite sensitive. So we have recording, so we record them, so we can check the time, the entry, and so we have a sophisticated recording system to do this. My collaborator did, and my student worked with them. So
okay, thank you.
That's the two that I presented is accepted model for cognitive behavior, at least for Alzheimer's. Yeah, people use this all the time. Yeah.
So maybe in the next speaker, Professor Lee will explain about the check the behavior in neonatal, prenatal mind. The second speaker. So I think this is all the questions from the participant. Yep. So I don't want to conclude this session. So it's very nice presentation, Professor Teruna. So if someone want to ask question to Professor Teruna, maybe they can send email to you. Is it possible?
Yes, possible. Siahaan@ku.edu. Just my last name at KU. KU is Kansas University.edu. Very simple.
So we will share to the all participants. So Finally, thank you very much for your nice presentation.
Thank you.
I hope you enjoy this meeting.
Yeah, I will stay as
you can
as long
stay
as I'm not sleeping.
because of this night time. Yeah.
Okay.
Okay. So now we move to, thank you very much. We move to second speaker, Professor Hee Jae. So before start the presentation, I would like to share his PPT, his CV, sorry. So can you see? Okay. Everyone can see?
Iya, Dok.
Okay, so Professor Hee Jae is a graduate from Department of Genetic Engineering from Korea University, and then got his Master of Science as well as also Master of Medical Science from Seoul, Korea, and then completed the Doctor of Philosophy in Kyuhee University, Seoul, Korea also, and then got the GSPS Ronpaku from Kyushu University, Japan. So there are a lot of experience related to the research fields. So nowadays he's a professor in Department of Pharmacology, School of Medicine, Kangwon National University, and also now he's stay in Jakarta as a research scientist in our department. So Professor Lee is the major fields of interest is mainly in neurophysico-pharmacology, pharmacogenomic, as well as also herbal medicine and medical education. So there are a lot of publication. The Scopus index is about 22. So that's all from the CV of Professor Lee. So time is yours, Prof. Lee, to share your presentation.
Okay, thank you very much, Dr. Wawaimuli, for nice introductions. And it's my honor to have a presentation in eForum Biomedika about my work. Today, I would like to talk about valproic acid, which has a mitogenic effect on glia precursor during the rat neocortical development. And also, I will show its autistic-like behaviors. Though it could be very unpleasant to look this photograph, it's about the Spina Bifida. As you know, Spina Bifida is a birth defect in which there's an incomplete closing of the spine and the membranes around the spinal cord during early development in pregnancy. Recent studies have shown that cortex abnormalities may also be present in patients with Spina Bifida. For example, frontal regions of the brain tend to be thicker than expected, while posterior and parietal regions are thinner. And thinner sections of the brains are also associated with increased cortical folding. Here you can see hot and cold colors. And hot colors indicate cortical thickness and is greater in the Spina Bifida group related to the control group. And on the other hand, cold colors indicate cortical thickness is reduced in the Spina Bifida group related to the control group. And still, we are not sure about the etiology of the Spina Bifida. It's believed to be due to a combination of genetic and environmental factors. For example, not having enough folate in the diet before and during pregnancy also plays a significant role. Other risk factors include certain anti-seizure medications, obesity, and poorly controlled diabetes. Especially valproic acid is well known an anti-epileptic drug which has Spina Bifida adverse effect. And valproic acid is an old generation anti-epileptic drug, but today is widely prescribed drug for bipolar disorder and migraine. And as I told you about Spina Bifida in the first slide, most importantly, BPA is associated with congenital malformations and development delays of infants who are prenatally exposed. This reproductive toxicity is a major issue to prescribe valproic acid to women of childbearing age, even though valproic acid is highly effective medications. Therefore, understanding the mechanism of teratogenic actions is very valuable to prevent the valproic acid adverse effects. The action of the valproic acid appears to be mediated by prolonged recovery of voltage-activated sodium channels from inactivations. Also, valproic acid produces a small reduction of T-type calcium cations and stimulate GABA synthesis and inhibit GABA degradations. Also, valproic acid shows Hsqc inhibitory properties, induce hyperacetylation of histone proteins, and it causes chromatin to unwind, such that transcriptional factors and RNA polymerase modulate the transcription of DNA. In 1997, valproic acid emerged as an anti-cancer agent because valproic acid inhibits proliferation and induces differentiations of neuroblastoma in vivo. Further studies have demonstrated that the anti-cancer effects of valproic acid are mediated through inhibition of class 1 Hsqc. Therefore, recently, valproic acid is used as adjuvant reagent in chemotherapy, especially brain tumor. Furthermore, Hsqc inhibition is suggested as a mechanism of teratogenic actions of valproic acid because it shows antimitogenic effects and induce cell death. Well, then can we say valproic acid is an antimitogenic reagent for its mechanism of Hsqc inhibition? The answer may be no, because previously it is found that prenatal exposure to valproic acid stimulates proliferations of neural precursor cell in rat4 brain. We injected 300 milligram per kilogram valproic acid twice a day subcutaneously to pregnant embryonic day 16.5 rats. Here in ventricular zone, and brown circle is the BRDU positive cells. And BRDU labeling index is increased in valproic acid group, which means valproic acid promotes S-phase entry in the frontal cortex. In cerebellar cortical neural precursor cultures, DNA synthesis is increased at 1 millimole concentration, but reduced at 3 millimole concentrations. Therefore, we conclude that BPA has a dose-dependent biphasic effect. These results are very interesting because previously people thought valproic acid inhibits cell proliferations, yet we have the opposite results. These data are focused on neuronal cells. So our next question is, how about glial cells? Why do we focus on glial cells? First, because glia are the most numerous cells in a human brain, outnumbering neurons by tenfolds. Well, neuronal cells is the majority in the rat brain, but within the cortex, glial cells are two-fold more than the neuronal cells. Therefore, if we study red frontal cortex, most neuronal cells will be glial cells, not neurons. And as you know, the astrocytes is the major cells in glial cells. Second, in fact, recent studies indicate that astrocytes are functionally diverse and play critical roles in neurodevelopmental disease, such as Rett syndrome and fragile X mental retardations. At an early postnatal stage, astrocyte promotes synaptogenesis and control of the synapse stabilizations and eliminations during their maturations. Therefore, a precise understanding of valproic acid effects on astrocytes could lead advance in understanding a variety of valproic acid defects during cortical development. So our aim of the study is to characterize valproic acid effects on proliferation and differentiation of glial cells, especially astrocyte on frontal cortical astrocyte precursor cells, both in vitro and in vivo. I'm going to break my talk into three parts about valproic acid effects on glia, especially astrocyte in vitro and in vivo. And final, I would like to present about outcome in the animal behavior. First, to study in vitro, a new culture system is required. Traditional primary astrocyte cultures takes a long time, more than a week, during which cells are incubated with fetal bovine serum. And such conditions may lead to modified cell character. Also, because we are interested in the developing stage, matured astrocyte is not our target cells. We want to examine valproic acid effect on maturing astrocytes. So we establish a new glial culture system. First, we dissect end of the olfactory bulb and take forebrain cortex on postnatal day 2 rat pups and mechanically dissociate and plate it in tea flask. And we incubated for only 3 days. There, the flask was shake overnight and the cells were replated in N2 media with BSA, which is a non-serum media. To demonstrate VPA effect on cell proliferations, thymidine or BRDU was treated for 2 hours before the next assay. And this is the result. First, we characterize the cells of new culture model. We're using nestins and vimentin antigens for neuroprogenitor marker and GFAP for astrocyte marker and TUJ1 for neuronal marker. And 24-hour replating, more than 99% of cells express vimentin and 96% of cells express nestin and 90% of cells express GFAP. Normally, more than 99% of cells were marked with GFAP in traditional astrocyte culture. And also only about 2% of cells are neuronal cells. That means these cells are non-neuronal cells. And it could be glial precursor cells, which are mature cells. Next, we want to know whether valproic acids could alter the cell fate or not. Only GFAP positive cells were significantly increased after valproic acid treatment. That means 24-hour treatment of valproic acid has not that much of change in cell fate. And using the new glial culture model, we perform thymidine incorporation test to confirm whether valproic acid can stimulate DNA synthesis or not. The results shown in dose-dependent thymidine incorporation data. After 24 cultures, thymidine was treated just 2 hours before doing assay. And valproic acid shows a dose-dependent biphasic effect in glia precursor cells. Especially clinically effective valproic acid serum concentrations has a maximum effect, and high dose 2.0 millimole of VPA group has similar results like control. That means 2.0 millimole VPA has a toxic effect or modulates extra signaling molecules. According to these results, we decide 0.6 millimole and 2.0 millimole concentration for further studies. Previously, we indicated that DNA synthesis was stimulated by valproic acid. Next, we treated BRDU to clear the valproic acid effect on cell cycle. Same as thymidine, incorporation test, BRDU labeling index is significantly increased in 0.6 millimole valproic acid group. And there was no difference in 2.0 millimole valproic acid group. That means valproic acid stimulates S-phase entry. Previous studies have reported that valproic acid interrupts cell cycle progression and results in cell cycle arrest. Even though valproic acid stimulates DNA synthesis and S-phase entry in our culture system, cell could be arrested in S-phase. Therefore, we count the cell number of cortical glial cells at 24 hours and 48 hours consecutively. You can see here that cell number are increased both after 24 and 48 hours. That means that the cells are consecutively proliferated at least 48 hours, and especially 0.6 millimole VPA conditions. Also, we couldn't find any difference in number of caspase 3 positive cell even in 2.0 millimole valproic acid group, which has no increase of DNA synthesis and S-phase entry. And this means 2.0 millimole VPA may not a toxic concentrations for glia precursor cells. So summarize in vitro study result, we established new in vitro culture system to characterize valproic acid effects on frontal cortex gliogenesis. And VPA may increase expressions of astrocyte marker, GFAP, and maintains expression of precursor markers, nestin and biomentins. Also, VPA stimulates DNA synthesis, S-phase entry, and cell number of cortical glia precursor in culture. However, effects are biphasic with no effect at higher doses. Therefore, now we can say valproic acid may stimulate proliferations on glia cells, glia precursor cells. But the next step is, but how about in vivo? We demonstrate whether valproic acid increases glia proliferation, especially astrocyte, also in vivo. We also established a new animal model because astrocytogenesis is peaked at postnatal day 2 in rodent. We inject 5 doses of valproic acid during P2 to P4. And thymidine or BRDU is treated for 2 hours before perfusions. Clinically, valproic acid promotes growth retardations and macrocephaly in some cases. Our data also presented that body weight and brain weight are decreased by valproic acid treatment. However, when we normalize forebrain weight to body weight, ratio of forebrain per body weight is significantly increased in valproic acid group. That means valproic acid treated forebrain might has more cells or larger cells than compared control group. As like in vitro, first we perform thymidine incorporation test at postnatal day 4, and 300 milligram per kilogram valproic acid treated group shows a 10% increase in thymidine incorporations in frontal cortex. This dose is equivalent of human for depressions model. And this valproic acid concentrations also effective on another animal model. And interesting point is, in rat serum concentration of 300 milligram per kilogram valproic acid after administration was between 0.2 to 0.6 millimole range. According to this data, we decided 300 milligram per kilogram concentrations to perform further experiments. Previous data indicate that valproic acid stimulates DNA synthesis in frontal cortex at P4. And next, we perform BRD immunohistochemistry and quantified total BRD positive cells in frontal cortex using serological methods. The green dots represent BRD positive cells in frontal cortex. And total number of BRD positive cells are 30% point increase in frontal cortex of BPA treated groups. And this result means valproic acid also stimulate S-phase entry in vivo. Then we like to know what kind of cells are BRD positive cells. We choose brain lipid binding protein, BLBP marker to identify the cells because it's known as an astrocyte precursor marker. You can see the majority of BRD positive cells are co-stained with BLBP marker, and about 80% of cells is BRDU and BLBP double positive cells. Also, number of BLBP positive cells are significantly increased in valproic acid group. Therefore, this data indicate that valproic acid stimulates astrocyte precursor cell proliferations in frontal cortex. And next question is whether those astrocyte precursor cells could sustain to live and mature to astrocytes. We treated BRDU for 6 hours after last valproic acid treatment at P4 and sacrificed the animal at P21. Even at P21, BRDU positive cells are increased in valproic acid treated group. And there was no difference of increasement of cell number between P4 and P21. That means astrocyte precursor cells sustains to live for differentiations to mature astrocytes. And we quantified total number of S100 beta, which is one of the mature astrocyte marker using serology method. And total number of S100 beta positive cells is increased in valproic acid group. Also, GFAP is increased in valproic acid group. And that means valproic acid-induced proliferating cells in astrocyte precursor and differentiated to mature astrocyte later. And furthermore, early exposed to valproic acid induced to the expression level of protein of glia cells, such as glia cell derived neurotrophic factors, GDNF, and excitatory amino acid transporter, EAT1. And summarize the in vivo results. Valproic acid reduced body weight and brain weight, but increased forebrain body weight ratio. And valproic acid stimulates astrocyte precursor cell proliferation in the frontal cortex. And the proliferated cells may survive and mature into the frontal cortex. And since there are no reports whether enhanced postnatal gliogenesis affects behaviors related to the neuropsychiatric disorder. So next we assess animal behaviors of postnatal valproic acid exposed group in adolescents. In detail, after VPA treatment during the postnatal day, PND2 to PND4, 4 behavior tests such as open field locomotor test and 11 plus maze test, 3-chamber association test, and passive avoidance test were performed at PND 21 and PND 22. First, to assess locomotor activity, we perform an open field test on PND 21. VPA treated rats showed 52% point increase in the total number of bar crosses, just suggesting hyperactive behaviors. Moreover, the velocity of crossing bar was also significantly increased by 59% in valproic acid treated group as compared to the control group. Those results indicate that hyperactive activity followed in valproic acid early postnatal exposure rats. An elevated plus maze test is well-known maze test to assess anxiety-related behavior. The preference for being in open arms over closed arms is calculated to measure anxiety-like behavior. We performed this test on PND 22. The number of the entire entries into the open arms and the time span in the open arm were both reduced in the valproic acid exposed animals than the control groups. And this result suggests that only postnatal valproic acid exposure shows an abnormal anxiety behavior. Next, to evaluate social behavior, we performed the 3-chamber social interaction test on PND 22. In the first sessions, we measured the sociability index, and in the second sessions, we measure social preference index between the novel and familiar rats. In the sociability test, the valproic acid treated rats showed significantly lower interaction times, reduced about by 45% point with conspecific rats. Furthermore, the valproic acid treated rats also spent significantly more time with the familiar rat and less time with the stranger. Both of those behavior measures suggest social interaction deficit following early postnatal valproic acid treatment. And the final, to evaluate the effect of the early valproic acid treatment on learning and memory, we perform a passive avoidance test on PND22, and no significant difference were found between the control and valproic acid treated groups. It means that early postnatal valproic acid treatment has no effect on this measure of learning and memory. So summarizing the animal behavior results, early postnatal valproic acid treatment induced hyperactive behavior and abnormal anxiety behavior and deficit in social behaviors. Also, early postnatal valproic acid treatments has no effect on learning and memory. Autism is a neurodevelopment pathology affecting 1 out of 100 children worldwide. And trauma and social consequences induced by autism are a real public health issue. As you know, clinically, autism is characterized primarily by communications and social interaction deficits associated with repetitive behaviors and restrict interests. The term of the autistic spectrum disorder, ASD, is used to account for the diversity of symptoms that characterize this pathology. And anatomically, the brain undergoes a period of precocious growth during early postnatal life, followed by decelerations in age-related growth in autism. And this figures is fitted growth curves of cortical thickness in autism and control group. And a graph of x-axis is age, and the y-axis is cortical thickness, and like a scatter plot around the periphery shows the curve fit to the cortical thickness data for each group at the vertex. As you see, there were significant group difference in cortical thickness between autism and control at the early age. And so in sum up, now we know about pathophysiology and clinical phenomena of autism. And for the study of autism, the question is, can we use our potential postnatal valproic acid exposure model as an animal model of autism? So, so the validation criteria are the essential values that are applicable to the evaluation of any animal model and are distinct by many ways. And one of the construct validity means that the model has a sound theoretical rationale, such as equivalence of a causative or triggering factor and underlying neurobiological mechanism. And the recent studies show that epileptic women who takes anti-seizure drug, especially valproic acid, during pregnancy are at more than double the risk of having children with autism spectrum disorder and nearly a double risk of having children with ADHD. And furthermore, prenatal exposure of valproic acid on embryonic day 12.5 is well-known animal model of autism. And in face validity means that the model has strong phenomenological similarities and similar pathophysiologies. As you see our result, we showed that postnatal valproic acid exposure animal induced brain enlargement and autistic-like behaviors. We therefore suggest that this new postnatal valproic acid exposure animal model may provide a new translational model of ASD or ADHD. And this is the first slide of the handout for my pharmacology class. And it means the right dose differentiates a poison from a drug. And might be valproic acid is one of the most appropriate reagents for this praise. And finally, thank you very much for your attention, and thank you.
Okay, thank you very much, Professor Lee, for the nice presentation. So I would like to inform to the all participant and the speaker because of the limitation of time. So we will combine the discussion session for second speaker and the third speaker in the end of after completed the third speaker. So it is okay. So you can also chat. Write your question in the chat box. So I hope Professor Ru will not leave this meeting. So can you able to wait for the end of the session? So Dr. Irawan, are you ready? Sorry, Dr.
Yes,
Irawan.
I am.
Oh,
Can
okay.
you hear me now?
Yes, yes. So, the third speaker is Dr. Irawan Satriotomo. I would like to share the CV of Dr. Irawan. Yeah. Okay. So Dr. Irawan is also our diaspora scientist from Gajah Mada University, if I'm not mistaken. So now he is owner, president, and the founder of TissuePro Biotechnical in US, and then also founder of Indonesian Neuroscience Institute since 2017, and also ex officio director of Indonesian Brain Research Center in Surya University. Dr. Irawan was get a Ph.D. from Kagawa Medical University in Japan, and then got the M.D. Medicine in University of Gajah Mada. So there are a lot of awards that he received, and then also the research support, and also many grant and a lot of publication. So without further ado, I'd like to invite Dr. Irawan to share his lecture. Time is yours, Dr. Irawan.
Thank you so much. Good morning, everyone. So because there is another speaker from South Korea, so I will speak in English, but you can asking the question later with Indonesian. So today I would like to introduce, and actually, by the way, that the title it was given by Dr. Wawaimuli, signaling pathway perspective in neurodegenerative diseases. As you can see here in the background, actually, so many signaling pathway. So, and then everything actually is interact with each other, can be inhibitors, can be also associate or accelerators too. So I will starting because I heard that this is for the student. So we will talking from the basic about the cell signaling. So cell signaling, or we can also call as signal transduction, actually is the transmission of the molecular signal from cell exteriors to the inside of the cell. So it's kind of the, if you get a phone call, you give a call and you call coming to your friend, and then there's the reaction of your friend. There is a communication between each cell actually. So the external signal is received and converted to another form to elicit a response. That actually the definition of cell signaling. So there is some general principle. First is signal act over different range. So there is a lot of cell signal, and every signal have their own target. And the second, and signal have also different chemical nature as well. And then the same signal can induce different response in different cells. So for example, we know that there's a couple, for example, the A2 is that people said it's only in the brain, but actually we found also in another part of the organ, like lung and heart, for example. So also the cell respond to set up the signal itself, and the receptors relay the intracellular signaling cascade. So there's a couple type of signal. First is the external microenvironmental that will given the physiological signals, like adjacent cell or extracellular matrix, cytokine, growth factors or hormone, glucose, amino acid, and others. And actually this talk will be focused more on external microenvironmental pathological signal, where we have the danger-associated molecular pattern or pathogen-associated molecular pattern, and also induce the inflammatory or immune cell on the system. The other thing also, the type of this signal is the internal hemostatic signal. I just given the talk last Saturday about the neuroplasticity, and I will touching base on this as well. So most signals are sent through the ligand binding to specific cell surface receptor, and then given the multiple extracellular similar to be distinct. So this thing, we can see that the multiple signal pathways actually integrated to make a response. So this is very complicated system that, as I probably you guys hear from a talk from Prof. Teruna, that there's multiple pathways that also interact to each other. So, for example, we have a growth factor, we have a tumor necrosis factor, cytokine, also others, hormone, or the other damage-associated molecular pattern. Then this is very multiple that can be interact each others. So the signal also can change the messenger RNA or protein level on the cells. That how they do with transcriptional activation or post-transcriptional microRNA stabilization. They can also do the translation control mechanism, or the post-translational protein can be destabilized or stabilized by phosphorylation or ubiquitination or acetylation. And the other thing is also the protein degradation, as Prof. Teruna mentioned, because when the signal coming in, or that there is some degradation effect as well. So the amount of the protein probably not enough to giving the effect, for example. So the other major player is kinase. We know that tyrosine kinase have more than 100 genes, like growth factor receptors or cytoplasmic, like JAK, SRC, FAC, and others. And also there is a serine kinase that is 400 genes, like a MIB kinase, TGF beta receptor or PKC, or also they have a dual specific kinase like a MAC. So this is the receptor tyrosine kinase. I can, as you guys can see, this there is a multiple factor from the epidermal growth factors, fibroblast growth factor, platelet derived growth factor or PDGF, insulin growth factor IGF, or neurotrophin salt like NGF, BDNF receptors, and NT3. So as I mentioned to you, this is really very complicated. So in this talk, I cannot talking all that kinase, but I will go to specific kinase and also specific diseases later on. So how about the phosphorylation? Phosphorylation is occur to alter activity of enzyme initiating cascade. For example, MAP kinase pathway initiated by activation of EGFR receptors. and also can be induced DNA binding like STAT or C-gene transcriptional activities. The other thing is changing the subcellular localization of the proteins and changing the protein stability. So the signal can act over different range. For example, for endocrine, it's a long distance. They come to the bloodstream like hormone, and then they go to the target cell. Or you can use like a local mediator like a paracrine. The other signal transduction is neuronal or synaptic. They use like neurotransmitters. Then also there's some direct contact cell-cell interaction like a delta or notch. So so how the signal respond? And first, the first messenger like ligands will be binding to the receptors and then make a cascade or a downstream signaling. This is what we call the signal transduction. Then can come into the nucleus and changing the gene expression or given the cellular response. So the others is ion channel receptors. This is the mostly in the synaptic, like nerve cell communication, but also can be happening in muscle contraction as well. So I will not talking more about that. So the thing is, there is a lot of student, and also every year mostly I going to Indonesia and giving a talk or giving the workshop. So there's a lot of question about how we can we see the or measure or manipulate the change in downstream signaling, and the answer is yes. If you have the correct antibody, then you can see the what is the signal, what is changing in the target of individual signal itself. So, for example, this is the work that I did in University of Wisconsin before. So we have what we call the respiratory neuroplasticity. So what we do is we anesthetize the animals, rat in this case, and we pump ventilate it, we paralyze, and we phagotomize. And then the rat will lying in the surgery bed, and then we control all the respiratory. So if we given the hypoxia, 5-minute hypoxia, the first bolt, and then we putting back to the 50%, and then we giving back again the 10% of the hypoxia, and we give the back to the normal until 3 times, and then when we putting back into the normal, or I mean the 50%, then you can see the increase of the amplitude of the phrenic nerve. This is we call the phrenic long-term facilitation or neuroplasticity of the respiratory system, and it's happening in the phrenic nerve. So if we do that, as I mentioned to you, then we have what we call the phrenic long-term facilitation. This is the plasticity that happening in respiratory motor neuron. How if we given the intermittent hypoxia sustains, meaning that we given the hypoxia continuously until 25 minutes. So interestingly, we see that there is no plasticity on the phrenic nerve. So then we continue, we have, this is interesting because our hypothesis is always changing every time. And then we have a lot of discussion about this. And we always try to put another molecule on our board. So the first the hypothesis that we thinking is if we did hypoxia into the animal, so the serotonin terminal will release the serotonin and will be uptake by the 5-HT2A or serotonin 2A receptor. Then it will induce the PKC to release the BDNF. And this BDNF will capture by the BNF receptor. This is very specific receptor for BDNF, track B. Yeah. And then previously, Prof. Teruna have been talking about this. This track B receptor become activated or phosphorylated and then induce the phosphorylation of the ERK that we will coming into the membrane and then induce the respiratory drive. And the other is AKT, and also will induce the phosphorylation AKT and coming into the nucleus and changing the gene expression there. So we will not talking about the protein phosphatase later on. So this is the data that we have been published a long time ago. It's like in 8 years. So if you see in normoxia and then if we given the hypoxia 3 times a day, so what's happening is the expression of the serotonin in the area of the phrenic nerve. The phrenic nucleus is located in C3, C4, cervical 3, cervical 4, and cervical 5. So I took this cervical 4 as a representative, and you can see that there is a lot of serotonin receptors that upregulated. Yeah. And we can, if we merge, we can see that. Then the location of the phrenic nerve actually is a very small area in here. I will showing you guys later on the detail. So how about the receptor itself? The 5-HT2 receptor also upregulated, as you can see, the normoxia. And then if we given the hypoxia 3 times for 10 weeks. So you see that there is a lot of upregulation of the 5-HT2A receptors on the phrenic nerve. This is the phrenic nerve. This is the nucleus, the cell bodies of the phrenics. And then how about the BDNF? Yeah, as again, actually, Prof. Teruna already given the talk about this. During the development, the growth factor or trophic factor is very high because it's needed for the brain development. But when we adult, we don't produce much of the BDNF. So except if we do the exercise as Prof. Teruna mentioned. So how if we exercise the rat with the intermittent hypoxia for a couple weeks, and then we can see that what you see in here is now become upregulated, and we found also with the colocalization with the confocal microscope, there's a colocalization between the cell with the BDNF. So it's also happening in its receptor, track B receptor, and we can see that the receptor also upregulated as well. So the normoxia also can induce the phosphorylated of AKT, and you can see that the phosphorylation coming into the nucleus and changing the expression there. So, and also if we see with the synaptophysin actually is the staining for the synaptics, and then you can see mostly is phosphorylation of the AKT is in the cell, not in the synapse itself. So the AKT again is coming into the cell and changing the expression there. And when we do the ERK, and we can see the ERK also upregulated on that phrenic nerve. This is very nice staining, and you can see that this is very specific localization of the phrenic motor neuron, and you can see the fibers coming out into the outside of the cell and giving the nerve to the respiratory, the major respiratory muscle is the diaphragm. So then also we do the colocalization, and then we can see that the pH are calculated in the cell, especially in the outside of the cell. And when we do the synaptophysin, and you can see there is a colocalization, so the membrane become very sensitive here. So the other thing also, as you guys see before, that the serotonin actually have an effect on the plasticity. So how if we bypass the system? We don't given the hypoxia, but we give the episodic of the serotonin, 1, and we waiting for 5 minutes, and 2, and we waiting for 5 minutes, and 3rd time, and then when we waiting and we see the plasticity. This is actually we can bypass the plasticity by given the molecule that needed for the plasticity. So the other thing also is the intrathecal, the BDNF. If we injecting directly into the phrenic nerve, we bedding them into the localization, the area in cervical 3, 4, 5. So we can see after 30 minutes, there is a plasticity. This is published by Becker Herman at our lab in Nature Neuroscience. So serotonin or BDNF itself can induce the LTP. Yeah, I mean the long-term facilitation of the phrenic. Yeah. So now I try to use the system to see how if we changing the system in the neurodegenerative diseases. The model that we use is amyotrophic lateral sclerosis. This is the primary motor neuron death, and sometimes people say it in United States is Lou Gehrig disease. This is Lou Gehrig is the Yankees players. He's very famous, and probably you guys also know one of the very famous scientists, physicists, Stephen Hawking. Yeah. The onset of the LS is 40 to 60 years, and the prevalence mostly in male predominance. And then this is actually very progressive, starting with the muscle weakness, then atrophy, then most of them dying because of the respiratory failures. Some of them using the respiratory equipment like the Stephen Hawking has. The problem with the LS is only 5 to 10% is due to the mutation of the supraoxide dismutase or SOD1. The others is like what we call the sporadic LS. We do not know what the cause is. So there is a lot of common theory like oxidative stress, excitotoxicity, protein aggregate, or mitochondrial dysfunction. So what we do is we try to use the mutant animal using the SOD1. So this is the LS from the human SOD G93A transgenic. So there is a couple different of SOD, G37, G85R, but we use the G93A animal, and we making them in our labs. So as you can see, if we matching between the mutant, we have the diseases with the others. So it will given the literate that actually the healthy one, but almost like 30% or 20% of them become sick and become have the disease. Then the disease is really very mimicking what's happening with human. So first they're starting with the paralysis, and atrophy in the hind limb, and then if you can see, they also have hard time to breathe. Why? Because of the phrenic motor neuron mostly die. So what we do is we try to see as you guys see before that this is the model that we have, the signaling model that we have, and then we try to see how animal can compensate that. Even though this is the wild type animal, you can see the phrenic motor neuron is really very healthy. There's a lot of motor neuron here. And when the animal become end stage or later on dying, so you can see all the motor neuron actually is mostly gone. And interestingly, when we try to see that how the animal behave, even though the motor neuron is already dying, the plasticity is still there. So meaning what? Meaning you guys have about 100% of the motor neuron, but with the 20% or 10%, you still can breathing normally. And also it's happening with our patient. But then because the motor neuron is become exhausted, you can imagine 100% is actually is need to do work, but now it's become only 10 neuron doing the work that must be done by 100 people, for example. So you can see that the motor neuron become very sick and then exhausted and then finally die. So if we given the SI, this is the RNA interference of the BDNF. Of course, we can see that BDNF is really necessary for the long-term facilitation. You see that it will be gone, the plasticity. But interestingly, if we given the track B, that even we try to challenging the receptor track B is still there, meaning there is something happening. There's some compensation mechanistic happening in this model. So when we also now this is again vehicle, you can see very dramatically the long-term facilitation of the phrenic is much more larger than normal. So meaning what? Meaning the phrenic motor neuron working very hard. So this is what happening in our model. So if we given the MEK inhibitor UO126, so again it's blunted. But if we give the PI3 kinase inhibitor, interestingly, is upregulated. So meaning what? Meaning there is some compensation. So our working model is changing again. So we're thinking there is a two system, what we call the hemostasis in plasticity. First is by Q pathway and the other one by S pathway. The Q pathway using the BDNF, MEK, and ERK, and the S pathway using the immature track B, PI3 kinase, and induce the AKT. So again, if we use the SI BDNF and then UO126, so this will be reduced. But actually, the other pathway, S pathway, is increased. So meaning that compensatory mechanistic that happening in our model, LS actually is Q pathway dependent. So the way that we're thinking when we try to give to try to find a good drug for specific specific diseases, this need to be thinking. So because there is a push or accelerator, then there is a brake, and both is like when we driving our car is actually is doing our hemostasis. So you guys need to know this thing. Yeah. So how if we do the intermittent hypoxia on that animals, I mean the ALS animal, we given the 3 times per week for 10 weeks with 5-minute interval, and we do the whole body intermittent hypoxia. So this is what's happening. This is very interesting data, and it's not published yet. So in wild type animal, or that given the normoxia, as you guys see it, there is nothing, because again, in adult brain there is the growth factor, in this case VEGF, is very low. But if we give the EIH in wild type animal, you can see the expression of the VEGF is upregulated. And then we can see if the mutant animal doesn't, we give the normoxia, meaning that is in the air, room air. So what's happening is there is still expression of the VEGF, very small cell is upregulated in here. So you guys know what's this. Dr. Lee just, Professor Lee just given the talk there. This is the small cell, actually is the glial cell. So meaning what? Meaning I always given the example, the easy example. So if someone in our neighbors die, so what happening? The neighbors from our neighbors, I mean the other people will come over to that people that probably the one of the members is died and given money, given food and everything. It's the same thing too. So the ideas of that glial cell actually is a very inflammatory cell. That's true, but in other side actually is good. Why they given the trophic factor to the dying neuron? So this is always become discussion when I giving the talk to the Perdosi, Persatuan Dokter Saraf Indonesia, to neurologists, whether is it necessary to give the anti-inflammatory for the neurodegenerative disease with the chronic situation. I don't think it is necessary. Then there is some data too, probably 5 or 10 years back, that for the ALS there is only one drug that approved by FDA, what we call the Riluzole. Riluzole actually don't do anything. They only given it's not expand the life expectancy, but it's only the quality of the life. There's a drug that have been done, have been studied to the ALS. I forgot the name. It's part of the tetracycline, minocycline. What's happening is there's no differences, and some of them actually die earlier because of this anti-inflammatory drug. So what we thinking that because you given the anti-inflammatory, so all that astrocyte or glia or microglia here is die, and then is exacerbated the process, and the patient is dying. So then how if the mutant animal, we given them with the intermittent hypoxia? Interestingly, this is actually the story that we have in our lab. We don't understand that animal that being taken care by our graduate student is living more longer, and most of them is living more than another one. Usually, by the way, the else animals die in a range of 120 to 130 days. But this animal that we give the intermittent hyperexia can live 150 to 170 days. So you can imagine it's given another 20% to 40%. If we can translate it to the human, so it's meaning we can extend it, the life of the patient. So yeah, this is still in our discussion. So, but we're thinking that probably this is an adjuvant to the patient that can be thinking in futures. So I will move forward, and then then what we're thinking is signaling pathway actually can be a tool as for the drug discovery, as Prof. Teruna mentioned. So as you guys can see in here, the neurological our CNS degenerations study using a lot of signaling pathways for these purposes. So one of them is mostly transcription factors, tyrosine kinase, phosphatase, and others. So I will try to talk a little bit of our study or our data that we've been done in a couple of years back. Is the JAK-STAT signaling, and then it's really related with neuroinflammatory diseases. So again, as you can see, this is very complicated pathways. So the signaling is because of the cytokines binding to the JAKs, and then JAKs will induce the STAT3 and then enters through the nucleus, changing the appearance of the PS or SUMO and other thing. And also it's related with also the SOX3 as well, that can prevent that. So we would like to try to see in our model, that the model that we use is a stroke model. So stroke is defined by interrupted the blood supply in the brain and resulting the tissue death and functional loss. This is become major problem in developed country and Indonesia too. So I also a consultant at Rumah Sakit PON, Neuroscience Hospital in Jakarta, and been discussed a lot of with the doctors there. Now we find a lot of young, like 30, 40 years old people is getting stroke. What is going on? Yeah, because yeah, we changing the lifestyle there. Yeah. So yeah, in US it's become third leading cause of death, as well as in in Western Europe. So and then there's no effective treatment. So the TPA actually still is become the first drug that can be entered to the patient, but still the result is not as we want. So this is the MCA occlusion in the person, and then if we use the fibrinolysis like a TPA, so it's get back to normal again. And we will use this model. What we do is we put the string into the middle cerebral artery. Yeah. So again, this ischemic. This is what we call the ischemic stroke. The ischemic stroke has a core and also penumbra, and core can be expanded, and then the penumbra become more core if we don't prevent this cascade. So this is what's happening with the stroke. So in the 6 hours, it will break the blood-brain barriers. Prof. Teruna has been talking about that thing, and also because there's this occlusion, so there's no oxygen on the brain, so there is the energy failure, and then it's starting with the activation of the glial cell, microglia, and cytokine will be upregulated as well. So this cascade is actually need to be stopped. So the important thing is, during the discovery of the drugs, so usually we use the animal model for these purposes. So I use the animal model, what we call the stroke model. So again, as I mentioned to you, that we will try to put the string into the middle artery. So what's happening is with the staining up, we call TTC, tetrazolline something. You can see that if the tetrazolline or TTC actually binding the mitochondria, so then you can see that it's really reddish. This is meaning that there is the oxidation on the tissue. So if the tissue is dead, then what's happening is the area of the brain is become white. So most of these models can given up to 40% of the ischemic. Yeah. So that in human you cannot have a stroke more than 40%. So yeah, mostly the patient will die if it's happening. So there's a the other small models, what we call that occluded system. So we just occluded for few, and then you can see that that's very small. It's less than 10% or percent. That is this model actually is mimicking what's happening in human. But the problem is this animal, this rat, after 2 hours, just walking like a normal. They're going to the mall or whatever it is. So we cannot use this model because we would like to have the model that we can see for a longer time period. So what's happening is what we do is we use these animals, the rat. So we have the sense animals. We just open, and then we see the carotid communis artery, and then we close again. So if we do this, of course, the animal is just SAM. So you can see that it's walking like a normal. There is no problem. And this animal that in the other side is actually the animal that we did for the ischemic model. And you can see this is a very, very big rat. This is like to almost 1.5 and 2 years old rat. Why we do that? When we apply the grant, the NI is asking us because most of the patient that have a stroke actually is middle age or like 40, 50 something. So there's no young people that get the stroke. So we need to have what we call aging animal as a model. So that what's happening is we have very huge animal and it's very expensive. One rat is cost us like 1,500, so 1,500 for animal. So again, nobody would like to touching except me. This is because usually I do operation like 8 animal per day. So for these purposes, I only can do the 4 animal per day. The reason is because most of the aging animal die later on, like 24 hour or 12 hour later. This is really very need to supervise and then really need to be carefully treated. So if you see this animal, you can see that the animals walking a circle because the front line is getting paralysis. So if I took up, you can see that the left front limb is getting paralyzed, so they cannot moving like that. That's why they do circle, what we call circling. So so we try to see what's happening in this animal. We do the operation on the animal, and we took the brain cortical, and we check it that, and we see the phosphorylated JAK2 in that animal. Then you can see that there is a lot of phosphorylation during the 6 hour, 24 hour, even 72 hours. Then you can see this area that we took for the pictures is really damaged. So the other ones is the static phosphorylation. Then we can see the similar thing too. There's a lot of static phosphorylation in the cortex and as well as in the striatum. So then we would like to see where's the phosphorylation is located. So what we do is we staining that brain, slicing brain with the ED1. This is the microglia marker. And you can see that in SAM, you cannot see that phosphorylation. But after 24 hours, you can see the ED1 is upregulated and positive with the JAK2. So meaning the phosphorylation of JAK2 is located on the glial cell. In this case, it's microglia. Yeah. And also we have another marker, OX42, because the reviewer asking us to do that. Yeah. And then we found that it's very similar, that phosphorylated JAK2 or OX42 is located in macrophages. So then we try to see how about the STAT3 following the transient middle cerebral arterial occlusion. We found a similar thing too. The phosphorylation of STAT3 is located in the glial cell, in ED1 positive cell or OX42 positive cell. And when we try to see in the NUN is the marker for the mature neuronal, so we don't see any localization. Yeah. And also we try to use the GFAP in this case, and then we don't see any localization too. So meaning that phosphorylation JAK2 is not located in the neuron or astroglia. And then we try to see about the phosphorylation of STAT3. And we found that phosphorylation of STAT3 located in neuron and reactive astrocyte. So then we try to use the adenovirus. In this case, we try to see whether the SOC3 actually can prevent of the neuronal death, as you can see in the signaling pathway that I showing you guys previously. So we found that actually when we use the control adenovirus, we can see that this is staining. I do believe using the regular I forget nasal staining. Sorry, yeah, nasal staining or crescent violet staining, and you can see there is some very pale staining on it. So this is meaning that there is some degeneration process on it. But if we inject it with the SOC3 with the adenovirus, we deliver directly into the brain before we do the operation. So it's prevent from the degenerative process. So then we try to see how the SOC3 can express in JAK2 and or STAT3, and we don't see anything. That's so meaning that actually the SOC3 can prevent the animal from degenerated using the transient middle cerebral arterial occlusion. So the next thing is, again, as I mentioned to you, we try to see whether we can prevent and in every single molecule that we found in JAK-STAT signaling pathway. So we use the AG490. This is the inhibitor for JAK, or we use the RNA interference for STAT3. So the interesting thing is we can also see using the SI glow. So we can see very nicely where is this injection is distributed. So one thing that you guys probably need to know, if the brain is still intact, meaning there's no degeneration process, there is no stroke. So all the injection site actually only in the injection part. But when someone or the animal got the stroke, so as I mentioned in the previous slide, the blood-brain barrier actually is broken down. So meaning what? Everything can enter. So this is actually the targeting process. I discussed a lot of data to my colleagues in United States or in Rumah Sakit Pon, because when we do this, we can injecting the drug delivery easily, because again the blood-brain barrier is already broken down. So for the drug, the JAK inhibitor, we use the aljet pump. So we putting into the intracerebral ventricular infusion with using the aljet pump. And for STAT3, we use the intracerebral injection directly. This is the data that we have. Then we also use the SI control for our control. So what's happening is, if you see that using the staining phosphorylated JAK or STAT3, you see the DMSO. DMSO is just a control for AG490. So you see a lot of staining up here. But when we use the inhibitor JAK2, so AG490, you see that phosphorylated of JAK3 is done regulated. Again, if we use SI control on that cerebral cortex, that image, so you can see the phosphorylated of the JAK2 and STAT3. But when we using the STAT3 RNI, so you see that expression is getting down. So when we try to do the this is actually not the best Western blot. Yeah. We found that phosphorylated JAK2 using the AMSO, you can see that expressed, but when we given the AG490, the inhibitors, the phosphorylated is not appear, as well as in STAT3. But if we given the SI control, so this interesting thing is phosphorylated JAK and is appear, but in STAT3 RNA is only appear in phosphorylated JAK2. It's meaning the STAT3 RNA is very specific just for the STAT3. So this is the data that we have with the AG490 and STAT3. We see that the reduction of the core and penumbra, and then also the expression of the neuronal death is also decreased. This is already published in Journal Neurochemistry 2006. It's a very old paper that we have. So we also seeing this apoptosis process. We use the tunnel staining, core and penumbra with the DMSO. You see there's a lot of staining on it, on tunnel positive cell. But when we use the AG490, this is reduced, as well as in the STAT3 sRNA. Yeah, this is the analysis that we done. So meaning the upregulation of JAK2 and STAT3 phosphorylation is occur following the transient middle cerebral artery occlusion, and this activation is of microcline macrophages is mainly showing in JAK2 and STAT3 phosphorylation. And there's a significant decrease if we use the JAK2 inhibitors or siRNA STAT3. So, and then this is the signal transduction that being investigated on the neurodegenerative diseases. Mostly people now try to investigate the PI3 kinase AKT signaling, and also check two-stat pathway also become a hot topic now. By the way, I found that JAK inhibitor as anti-inflammatory is been approved by FDA. So this is company. I'm sorry. I think this is already approved by FDA. So Celgence or Tofacitinib, and also baricitinib and upadacitinib is already approved for the FDA to treat the rheumatoid arthritis. The question is, if can we use this for the stroke? So again, as I mentioned to you, that it needs to be done properly. We need to have a data and everything. So, but meaning what? Meaning actually a lot of drugs that have been used for other purposes now given use for the neurodegenerative diseases. For example, Parkinson's disease. People using for anti-epileptic CNS stimulation or anti-cancer, anti-asthma, amyotrophosis. This is also some of them using them antimicrobial, anti-epileptic. So a lot of people try to using the multiple method, because again, the pathway is not only just very simple pathway one, but also there is a lot of interaction between each others. So this is the model of candidate pathway contributing neurodegenerative disease. So a lot of like aging, development, genetic, epigenetic, systemic environment like inflammation process, vascular factors, or local tissue environment like a cell addition. And Prof. Teruna has been talking about how blood-brain barrier is so tight. So there's a lot of factors that's going on. That's why the disease become very hard to treat it, because again, very few, even small molecule, I just see that only below 500 Dalton can enter that. Then most of protein and also monoclonal antibody or drug itself is above 150 kilodalton. So it's really hard to enter that. And also there's an intracellular mechanistic as well, like apoptosis, autophagy, or mitochondrial function and other things as well. So what we do now? So there's a lot of we have a genomic, we have transcriptomic, we have proteomic, we have metabolomic, and a lot of things need to be done appropriately. This is need to seeing as an integrated process when we do the drug discovery for neurodegenerative diseases. Again, this is my last slide, probably. This is our postdoctoral fellow. Now some of them is back to their country or become assistant professor in some university. I work with Dory Sylvain, building the city R&D center there. And then our collaborators, Randy now moving to Harvard University, Julian still in Saskatchewan, and Clive in Cedar Sinai for stem cell. Yeah, I don't talk about stem cell probably in the next slide. Okay, that's probably that last slide that we have, and I will be glad to entertain some of you guys' questions. Thank you.
Okay, thank you very much, Professor Satriotomo. Very interesting speech. So now I would like to open the discussion session. So first of all, I would like to read some questions for Professor Lee. Professor Lee, are you here? Oh, okay.
Yes.
Okay, so this is the first question from Kenny Chantika. So you said that the administration of valproic acid at the dose of 300 milligram per kg body weight will decrease the whole brain, but the forebrain is increased. So can you explain this kind of phenomenon?
Okay, thank you for the questions. And well, I think the effect is not directly direct effect of the valproic acid, because when I expose valproic acid during postnatal day 2 to 4, so after exposure of valproic acid, the pups looks very stabilized for a while, and it means, well, it's compared to the control, I think there's some lack of nutrition, because the control is much more activated and it has much more chance to take it from their mom, so them. So I think one of the reason is the less of the body weight and less of the brain weight is might be is effect of the lack of the nutrition. And another issue is one of the adverse effects of valproic acid is weight gain, because it initiates pancreatic insulin secretion. So it's well known that after treated valproic acid, some patients have the weight gain. So I think the losing the body weight.
So I mean, I think this is mistaken. So they ask not the body weight decrease, but the brain weight. The brain weight is decreased. Is it true? The brain.
Yes, the body weight is decreased. Also, the brain weight is also decreased, depend on the body weights. But what I showed it is the ratio. The ratio is increased.
Okay. Yeah. So the second question related to the cognitive assessment. So apart of passive avoidance test, is there any test we can do for assess the cognitive function in the neonatal rat?
Well, the neonatal rat means less than postnatal day 14, right?
So you perform the cognitive test in adult rat or in neonatal rat?
Because I say it's adolescence, because I do on postnatal day 21. So, but as you know, before by postnatal day 14, they still close their eyes, so they cannot move along. So
Okay.
hard to do the behavior test.
Oh, okay. So the postnatal means only to give treatment, yes, but the test is after the adult.
Yes, not adult, but it's around the postnatal day 21. Is about like adolescence.
Oh, okay, yeah. So got it. So next question from Riti Muhammad. So could you explain more about the how valproic acid administration could lower the total mass brain? Is it for specific in frontal cortex? What factor that could make it happen?
I say the answer that before, because I think that the reason is the lack of the nutrition.
Next one is their interest in the proliferation and maturation effect of astrocyte in postnatal model. How long valproic acid administration to maximizing proliferation in animal model? Is there any factor that inhibits astrocyte proliferation?
It depends on the dose, concentration of valproic acid, because when I inject 200 milligram per kilogram, there was no effect. And in case of 400 milligram per kilogram, like most of the pups, like about 50% of pups were died after exposed to 400 milligram per kilogram. So it's very, it's very hard to handling the dose. So. And also, if you inject more high concentration, I think it has the antimitogenic effects of valproic acid.
Okay, got it. Next question is about also valproic acid. Could valproic acid give abnormal proliferation of glial cell into the neoplasm process?
Yes, because today we use it for the adjuvant therapy of brain tumor right now. So we can use valproic acid for inhibit the proliferation of the glioma.
I see. Okay, so maybe this is the final question for Professor Lee. From Rizmi Diana. So can you explain why there is no difference in learning and memory retention in early postnatal valproic treatment?
Actually, well, we didn't see the hippocampus, so I have no data of what kind of change or modifications are occurring in the hippocampus. But what we do the PESO avoidance test, there was no difference, and so okay, because most of the autistic patient children has no difference in the memory, so we see it's okay, then keep going on the next behavior. So I'm sorry to answer about that.
Okay, thank you. Next, as I know, autism disorder came not only from the overgrowth of the cerebral cortex, but also in amygdala. Is it possible that valproic acid can affect glial or neuron cells in amygdala too?
I think it depends on the exposure date, because when we expose on the embryonic stage, it could affect on the neuron. So, but in our case, we inject in postnatal day, so I think it's affects most of this in the glycolysis.
Okay, got it. Another question from Sarsanti from Imeri. So, I was wondering about the behavior study after valproic acid treatment. What is the indicator to determine hyperactivity and abnormal anxiety by behavior in the animal models? Indicator to determine the hyperactivity and abnormal anxiety in animal models.
So piece of hyperactivity is, in my case, we measure the total crossing and the velocity. So vector to measure the hyperactivity. And for anxiety, we did elevated plus maze, and we can measuring the open arm increase or the time of spanning the open arms. So if they span less of the open arm, we can see it has anxiety.
Okay, maybe this is the last question for you. Also from Sasanti, valproic acid treatment induced the gliogenesis to stimulate cell numbers of cortical glial cell. Is there any correlation of glial cells and calcium or GABA expression in the brain?
Well, it could be because when we see the expression glial protein, well, we show that the GDNF or EAT1, so it's a excitatory amino acid transporter, so it expresses more, so might be GABA Well, we did not check about the GABA transporter, but might be GABA transporter also express more than comparing to the control. So.
Okay, thank you, Professor Hee. I think this is all the question. Thank you. You answering well. And then next, Professor Satriotomo, Dr. Irawan, maybe you can see in the chat board by yourself because of I'm confusing. Okay. So
okay.
I think yeah. So the first one from Husnul. He asks about the sorry, I'm mistaken. Oh, the first one is from Yudi Wono Wiwoho, Dr. Irawan. He asks about, I would like to ask about the signaling pathway for the neuron regeneration after brain injury or after hemorrhagic stroke in brain. Is there any biomolecular process found that can improve the neuron regeneration in brain?
That's very good question, sir. So yeah, actually we try to investigate it again. I don't talk about more about the stem cell or cell puncha in this case. So actually in our brain, in a supraventricular zone, and also the indentate gyrus of the hippocampus, we have the precursor cell, but the number is so small. So and then when we got the stroke, so they will migrate with using the radiant glia cells, so they migrate to the stroke area, but the number is very small, so it's not enough to regenerate the process. So what's happening, that's why now the stem cell it's become hot topic where we can replacing the cell that already died. So that's what we do. But again, one thing that I always discuss and I was confront the people, how can you put the cell in the very hypoxic environment like a stroke, and also there's very hyponutrient too. So what I did actually with Clive Spanson at UCLA unit in Cedar Sinai, he's the director of the Cedar Sinai regenerative medicine there. So we developed what we call that the stem cell induced trophic factors. So meaning when we put the stem cell inside that area, so they can also release the trophic factors, and then they can regenerate and then proliferate, and then also changing the profile of the cell. That's what we do, but it's still on the preclinical step. That's I cannot see showing you the data yet. So the other thing also, we try to using the trophic factor as I mentioned to you before, and also the doctors Prof. Teruna also mentioned. The trophic factor actually is very necessary for the brain to develop. So, but the problem with us in the developed brain or the aging brain, we don't produce more of the trophic factor, BDNF, VGF, IGF, and everything. So we need to have that inside. What we can do that we can supplement like what the Prof. Teruna explained before. But the problem, because all the protein is really very huge, so this is the problem. So we cannot do that thing. So this is still ongoing investigation, but I'm very lucky today. I hear the talk of the Prof. Teruna. So probably the adjuvant of the ADC5 can helping to helping us to putting back the trophic factor that we needed to induce the proliferation of our progenitor cell in the brain. Yeah. Probably that's what I can say right now.
Okay, thank you, Professor Irawan. So next from Husnul, FK Brawijaya. So thank you for your nice presentation. Does neonatal ischemia has the same pathway to intermittent hypoxia regarding to neuroplasticity? And what is the therapeutic challenge?
Can you repeat again the question? I'm sorry, I cannot.
Does neonatal ischemia has the same pathway to
Wow, yeah. Okay, Husno actually is my student when she taking the PhD program. So this is a very good question. Actually, is the hypoxic neonatal is totally different. Yeah. So it's not the intermittent hypoxia because it's sustained hypoxia. You get repressed all the time. So there is no plasticity going on. But again, in neonatal, we know that the brain is still developing. So then in a couple, when they develop, they try to compensate. I just given the talk last Saturday about the plasticity, neuroplasticity. And the neonatal brain actually is really very plastic. For example, I give you the hydrocephalus kit. So some of them can have the effect, but some of them can regain if they have a training properly. So meaning that the brain in the child or neonatal is really very plastic compared to the adult. So so the answer is neonatal hypoxic is not intermittent hypoxia.
related to therapeutic challenge, Professor? Therapeutic challenge.
Okay. Wow, this is a good question, sir. This is when I was working with Yohana Surya at Surya University. Actually, I got the grant 2 million rupees, 2 billion rupees, 2 million. But unfortunately, the center and also is from the Pangdam Jaya at the time. They would like to build what I call that mobile intermittent hypoxia. So meaning that we can bring that intermittent hypoxia tubing everywhere. So the ideas actually when I presented into the Pangdam is right now what we have a big chambers and then we need to adjust it everything. But actually, I with helping from one of my colleague in MIT Indonesian person actually. By the way, I would like to mention we have very smart people, Indonesian smart people everywhere. But our government sometimes or our people doesn't appreciate that. Now that he fly away to Malaysia to work with. But my point is when we discuss that, we can put the oxygen tubing and then we put together with the chip of the computer and we can make it that intermittently delivery to the, and we use the mass. I use the pilot mass. And then we can deliver that. So actually it can be therapeutic. So that the ideas that I given to the Pangdam is this is actually for enhancing our soldiers. But in this other side, actually we can also treating the patient as well. So because it's mobile intermittent hypoxia chambers, so we can using everywhere. So and then I have question from I forgot, is from Navy officers and I forgot the brigadier from Navy, and whether we also have the what we call the hyperbaric chambers, whether hyperbaric chamber can be used intermittently and giving the effect of the similar effect. The answer is yes. One of my graduate student, the postdoctoral fellow actually doing this thing. And then we found that it's very similar. So the interesting point that I would like to mention is intermittently actually giving more effect of the plasticity rather than continuously. Yeah. Continuous intermittent, continuous hypoxia or continuous hyperbaric. So there's no effect at all of the plasticity. But if we given the intermittently, then we have it that effect. So yes, the answer, we can use the intermittent hypoxia as a treatment to and actually we have been if you who's the name Randy Trambowers now is moving from Emory University to Harvard Medical School. Actually used that. He came to me and then asking whether that that intermittent hypoxia can be used for the spinal cord injury patient. And I said that, why we don't try it? Yeah, and he tried that. Now, actually, he got a very large grant, and then we found that spinal cord injury patient actually get very benefit from the intermittent hypoxia. Usually, this patient have been treating by neurologists for years. Most of them is the soldier from Iraq, Iran, and then they cannot do anything. And you know that this causes $1 million problem for our government, U.S. government. So then we try to use this as a novel method to treat the patient, and then the result is stunning, because we can see now they can stand, and then some of them can walk again. Now they can walk more further. So how is happening again? The intermittent hypoxia actually can induce, as you see in my slide, brain-derived trophic factor, vascular growth factor, and other growth factor that's necessary for the development. I think that's my answer.
Okay, very clear explanation. So it is a question from Kavin. So he asked about the role of circadian clocks gene regulating the respiratory neuroplasticity of phrenic motor system. I'm not whether it is pronunciation.
Okay.
Phrenic motor system. Yeah.
Very, very good questions. Whether the circadian have an effect on it. So the answer actually is yes. This is a story coming to our lab too. There is a graduate student, M.D. Ph.D. program student that come to me and asking, what the best time to give in the intermittent hypoxia? We know that the rat actually is nocturnal animal, don't they? And we are diurnal. We acted during the daytime, but the animal is active during during when we sleep. So that's why in the lab, so you used to adjust it that time. So what he do is he try to do the plasticity during the daytime, then the diurnal time, and also the nocturnal time. He came in like at 2 or 3 p.m. and then doing the research there. But the interesting point in his data that is not published data yet. I encourage him actually to publish it, but he already become a doctor now that doesn't want to do the research. But anyway, so during we do the activity, actually is the major the most prominent time. So yes, the answer is, do you know the circadian actually have an effect on our plasticity in general, not only just so. What is the best time? This is probably you guys want to know. So the best time is actually when the animals starting to do the activity. So it actually is around 4 to 5 to 6 p.m. This is probably why God, in our religion, try to get up early, do your activity, because it's the best time. And then our plasticity is showing the same thing too. Our plasticity double compared to the when we do it actually in other time. So yeah, probably this is answer this question.
Yes, very clear explanation. So next from Santi from Imeri. Nice to meet you again. Santi asks about she will going to have study about the neuroplasticity in diabetic mellitus and depression patient. Of course, it is hard to examine the BDNF in brain tissue from patient. Is it possible to check BDNF in serum samples from the whole blood? I mean, BDNF in serum can represent the concentration of BDNF in the brain. Is it correct? And then a second one is how is to differentiate morphologically microglia and macrophage cell in the staining method in your study. What is the related function between microglia and macrophages in human system?
Okay, this is also a very good question. Yeah, Santi always helping when I am at email. Yeah, he's always entertaining me there. So the concentration of the BDNF in our serum is very low. Oh. That's why sometimes we cannot capture that thing. So, and then the other thing also you guys need to know that that actually probably I need to rephrase again. The BDNF that floating in the serum actually is the immature BDNF. We have the immature and mature BDNF, and that whether it's representative, I don't know. There's probably I can sending the Santi later on. There's a couple of papers because the BDNF, if you have very good skills, you get to have a good tools. Yes, you can measure, but the measurement is like a picogram, very small. So that if it's sensitive enough, so you can measure that actually whether that is representative with the BDNF in the brain, we don't know. Yeah, but we can measure that. Then actually we can also comparing between each other. Last time when I went to Imeri, actually this discussion is also happening there as well. How we measure the BDNF in I think this is Rina. Dr. Rina Imeri for the nutrition director there, so we discuss about this thing too. So yeah, you can do it, but again, you need to have a good skill and good tools for measuring that. And the second one is about the microglia and macrophages. Yeah. This one is two distinct. The microglia is actually the cell that already located on our brain, and then can become proliferated, what we call the resident microglia. So they can become activated when we have damage or there is some stimulation there. And macrophages actually is not the resident cell in our brain. It's coming from our blood or our capillary. So when it's happening, that will enter to the brain tissue from the capillary system because it's broken down or it's damaged. So this is the two things. So then the question is, how about the markers? This is still a lot of debating. If you see my paper in Journal Neurochemistry in 2006, so I used the ED1 because we thinking that ED1 actually expressed in both microglia and macrophages. But then the reviewer said that this AD1 actually is more expressed in the macrophages rather than microglia. That's why we use the OX42 to microglia. So yeah. So that both actually again what the function in the brain. Yeah, they are the function of the microglia and macrophages is to cleaning the cell that the dead cell in the brain. So that's why you see that they are very active. You can see they have become large and then eating the debris of the cell there. So but again, as I showing the picture before, there is some microglia actually also express the trophic factors too during the disease progression. Yeah, I have the data, but I cannot show it because it's not published data yet. Yeah.
Okay, thank you very much, Professor Satriotomo. So the last question is from Professor Ina. So maybe Professor Ina can ask by yourself. This is related to
Okay. Thank you very much, Dr. Irawan, such an inspiring lecture on talk. So this is because we would like to know, since you are the founder and president of TissuePro Technology Corporation, can you please tell us your experiences and also challenges being an entrepreneur and also a pharmaceutical company in the US. So you are an entrepreneur and also a scientist there.
All right.
That this opportunity is open for them to be a scientist and entrepreneur even abroad. Thank you very much.
All right. Waduh. This is very, very private question, probably, yes. Okay. Okay, during my career in United States, I was working in Japan as an assistant professor and moved to United States. And then it's very different, the environments. But I have a lot of connection or a lot of experiences working with the pharmaceutical industry. One of my two of my students in when I was working as an assistant professor in Japan is actually working at Takeda Pharmaceutical and also Taiso Pharmaceutical, two the biggest pharmaceutical company in Japan. And yeah, I supervised them, and then during that time, actually I have a couple different patent. I call patent because I don't want to patenting in Japan. And then when I came to US, I'm very busy working as a research professor there and supervising. I have no time. So then when I am thinking that if I can use these experiences that I have in Japan and also in academia, and then putting together and building the good company that can providing the good drugs or good solution for the people that will be good. Because the problem in academia sometimes it's very hard to moving forward. Because first you doesn't have grant enough, and the second one, the environment in academia is really hard. For example, if I'm working there as a trainers professor, so all that activity that I did in outside is a part of the university because we have a contract. So then I'm thinking that I'm very sorry to say to my graduate student, my postdoc. So I said that I need to moving a little bit further and only just working as an adjunct professor in UF. Then I starting to build my own company. Then the good thing with this situation is I still have a connection with the University of Florida, University of Wisconsin, UCLA. So I use this connection to selling my product or doing the research together with them. I still doing the research and I still supervising some of them. But in other side, I still developing my own product. One of my product actually I call the biothermal. So you guys know that until now, even in United States, when organ is transplanted, so the organ donors is taking out and then have to our window to transplant to other people. So they only put the heart or in the ice bucket, very old methods. So what I do is I develop what we call the biothermal soul. So this biothermal soul is a solution. I'm sorry I don't showing that. Probably next time when we discuss about the entrepreneurship, probably I can showing you that actually can prevent the cell actually contain all. This is the keys, trophic factors, and solution, and also the energy for the tissue. So then when I put that kidney or the heart or whatever in that, can expand it, the lifespan of the organ, expanded that to the human. The problem with the human, as I mentioned to you, that the rat is very small. So meaning the perfusion of the tissue is really easy. But if you have the heart like this, the perfusion is really very hard to enter all the system. So I need to have Teman to see whether we can working together to develop this. If is happening, is will be breakthrough for the treatment for the transplantation organ, so it can extend it. And so that's what I'm do. By the way, Dr. Lee, I have a distributor in Korea. So if you want the product, our product, you can asking them to provide that solution. And this solution actually can also extend it the stem cell. So I use that for the stem cell purposes too. So usually you guys using the DMSO. And when you take out that stem cell from the tissue, is already 40% or 50% is die, because it's just the MSO, and it's because it's freezing, freezing fractures. Yeah. But with my solution, the tissue actually can living more, because even though you put in -80, this is not freeze. That's why I call anti-freeze solution. This biothermal can extend that. So then it can give them the energy as well. So in global, I said that H got the grant from the government in as well as in state, so then I can continue this. So yeah, I encourage you guys to thinking about that. And by the way, you guys ever heard the Gatorade, the drink? The sport,
like
Pocari Sweat. Like Pocari Sweat or something like that. Gatorade is developed by the doctors in University of Florida. It's $1 billion revenue. Now they have like 5% of the $1 billion. You can imagine now that the doctors. So yeah, if you have an ideas, you have saying that actually it's not only just academia. So you can also go to the industry, or you go others. Yeah, I think that.
Thank you very much. Thank you very much, Dr. Irawan. Thank you very much. So we are looking forward for your next talk about entrepreneurship. We will invite you again to talk more about that, because we need also help to develop the entrepreneurship for our scientists. Thank you very much for your inspiring talk. Thank you.
Okay, so because of time is up, so we have a limitation of time. So maybe I will close this session. So thank you very much for the participant and also for the speaker. So I move to the MC.